Our study was a comparison between different sites of lung cancer, primary site, lymph node metastasis, and distant organ metastasis, and the differences that are encountered when profiling these tumors at different sites. So we have noticed in our series of cases, which at Caris Life Sciences includes more than 10,000 lung cancer specimens, we noticed that they were coming from different sites. So the specimens that were sent to us for the evaluation could have come from primary lung tumors, could have come from their metastases in the lymph nodes, or they came from various distant organs. So what we tried to identify, what we saw, is that there was a difference in profile from these tumors when they're coming from different sites. So we collected all the cases and run a statistical analysis, which indicates indeed that different sites of lung cancer would have different profiles. What at that time we couldn't say whether that is because there are different patients who have been submitted just from the different sites, or is that a general phenomenon that occurs with each cancer sample that we get. So our next step in the study was to select for patients that had matched samples. We identified about 150 such patients in which we had matched or paired samples from primary site, from lymph node metastasis, and from the distant sites. And then we looked at the comparison the same way we did that unmatched large cohort and came to the same conclusion. So the main study outcomes appear to be that tumors collected from different sites and different times show differences. So there are spatial and temporal differences in biomarker expression between sites and samples of lung cancer. And those differences sometimes mean that therapy associations made with these biomarkers do not hold the same throughout the disease progression, indicating an issue with heterogeneity of the tumors that is both spatial and temporal. These studies are absolutely revealing that we cannot just rely on a single biopsy of the patient, and we cannot rely on a biopsy that is too old. When therapy can induce changes, selective pressure that is exerted on cancer progression while chemotherapy is given and the resistant clones emerge, cannot be properly treated based on the samples that were tested in the past. So resistance emerges for various reasons. One of the most important reasons is the heterogeneity of the tumor to start with, but also the evolution of the cancer and cancer populations during the therapy. We certainly need to modify the way we profile patients to include as many as possible sites and as well as samples that are collected and profiled just before the next therapy is planned. We also need to look at the different methods in order to comprehensively evaluate biology of the cancer as it emerges from previously treated to a recurrent and less responsive type of disease. So new methods are certainly needed. Again, those methods should be able to address the prediction of the therapy response, not just based on the biomarker that's present, but the way the disease behaves.